Today we'll be doing strawberry DNA extraction. You'll need a variety of supplies: coffee filters, plastic bags, a skewer, measuring spoons, strawberries, they can be frozen or fresh, dish soap, salt, rubbing alcohol, ice cold, rubber bands, small cups, and some water. If you have some of these more scientific supplies, you could use test tubes, snap caps, pipettes, cheesecloth, and scissors. You'll have all your supplies with you on a table. Next, you'll take the Ziploc bag and take your strawberries and put 3 to 4 in the Ziploc bag. Next, you're going to want to take all of the air out of the Ziploc bag and start smashing the strawberries. While smashing the strawberries, you're going to want to make sure any big chunks are smashed as small as they possibly can be. The DNA is located inside the nucleus of the cells, which means you need to manually break apart the strawberries to break down all the different cell walls and the nuclear membrane. Continue smashing the strawberries until there are no chunks and it kind of looks like a smoothie. Next, you're going to want to take your lysis buffer solution, which is 2 teaspoons of dish detergent per 1 teaspoon of salt and half a cup of water. You're going to want to add about 4 tablespoons into your strawberry smash. Then take all the air out of the Ziploc again. And then you're going to start mixing the strawberry solution with the lysis buffer solution, making sure to mix it gently so you don't get too many soap bubbles. The soap and the salt in this solution help clear away all the other lipids and proteins and break down the rest of the cell membranes, getting the DNA ready to be extracted. Next, you're going to get the cup and cheesecloth ready to strain the strawberry solution. You'll put the cheesecloth over the cup, push in the indent a little bit, and then take a rubber band and put it over the cup. Make sure once the rubber band is over the cup that you make sure to push a little indent into the cup again so the strawberry solution has somewhere to sit. Now you will pour the strawberry solution carefully into the cup with the cheesecloth over the top and allow the liquid to strain through to the bottom. The cheesecloth will collect all the bits of strawberry on the top of the cheesecloth, and the liquid that goes through to the bottom will hold the DNA in the liquid solution. Once the solution has drained through the cheesecloth, you'll carefully take off the cheesecloth and the rubber band, holding them above the cup to make sure the strawberry chunks do not fall into the solution below in the cup. Then take the cheesecloth and strawberry chunks and put them into your Ziploc to throw away later. Next, you're going to take the strawberry solution that's in the cup and pour it into the test tube about 1/3 of the way up, or about 5 milliliters. If you do not have test tubes, you can leave the solution in the cup for the next step. After the solution's in the test tube, or if you leave it in your cup because you don't have a test tube, you're going to want to get your isopropyl alcohol or rubbing alcohol out of the freezer because it needs to be cold. And then if you have pipettes, you can take your pipette and put about 10 milliliters or double the amount of strawberry solution into the test tube. If you only have a cup, you can slowly pour the rubbing alcohol into the cup, and the strawberry solution and rubbing alcohol will stay separate, just as they will in the test tube because of density differences. Once the rubbing alcohol has been added to the cup or the test tube, you can watch what happens. You will slowly start to see bubbles coming up out of the strawberry solution up into the alcohol solution. You'll start to see bubbles coming up, and those bubbles will have white stringy mucus attached to them, which is your DNA being pulled up into the alcohol out of the strawberry solution. Once you see a good clump of the stringy mucus coming up, you will take the DNA and pull it out of the test tube or out of your cup. This is where you'll use the skewer. You'll take your skewer, put it into your test tube, put the skewer against the side of the test tube, and pull out your DNA chunk. Depending upon your previous steps, you'll have either a little bit of DNA or really huge chunks of stringy mucus, which is your DNA, coming out of the alcohol. One reason strawberries are really good for extracting DNA is because strawberries are octoploid, which means that they have 8 copies of the DNA for each chromosome. Humans are diploid and only have 2 copies. Thanks for spending time with us today and learning how to extract DNA from strawberries. We hope you join us for the next video. When you do this activity at home, feel free to take pictures or video and tag us at EPSCoR. Tell us all about your fun with STEM at home. Feel free to check out EPSCoR's website for more activities that you can do under our Education tab or our Nature Sunday Academy tab.